One of the highly tested topics in USMLE are pyruvate and TCA cycle. If you understand it, you will unlock most of the questions regarding metabolism of UWorld and NBMEs. Hello everyone, it's Dr. Mohib, and in this lecture we will be going through pyruvate metabolism, Cori and Cahill cycle, pyruvate dehydrogenase complex, mitochondrial disorders like pyruvate dehydrogenase complex deficiency, pyruvate kinase deficiency, TCA cycle, and at last, some memory drills. First of all, pyruvate metabolism. During glycolysis, a single 6-carbon molecule of glucose is converted into 2 molecules of 3-carbon pyruvate. If we refer to the glycolysis, a single glucose molecule is converted into 2 molecules of pyruvates. While doing so, in this step, during the conversion of 1,3-biphosphoglycerate into 3-phosphoglycerate, we have conversion of NAD into NADH. So during the conversion of glucose into pyruvate, we have conversion of NAD into NADH. This disrupts or increases the ratio of NAD to NADH. NAD plus is required for normal continuity of glycolysis or conversion of glucose to pyruvate. This was all about pyruvate. Now let's go through the fate of pyruvate. Pyruvate can be converted into following 4 structures. First of all, it can be converted into alanine through Cahill cycle. Number 2, it can be converted into oxaloacetate. Oxaloacetate is a 4-carbon structure. Pyruvate is a 3-carbon structure. Alanine is also a 3-carbon structure. Then it can be also converted into Acetyl-CoA, which is a 2-carbon structure through Pyruvate Dehydrogenase. It can be also converted into lactate through Cori cycle, which is also again a 3-carbon structure. So first of all, we'll be going through the number 3 or Pyruvate Dehydrogenase. It is the most evolutionalized pathway. The pyruvate or this whole process conversion of glucose into pyruvate occurs in cytoplasm of the cell. Now for conversion of pyruvate into acetyl coenzyme A or CoA, the pyruvate enters the mitochondria and the enzyme pyruvate dehydrogenase complex converts it into a structure acetyl-CoA. By doing so, a molecule of CO2 is liberated and NAD is converted into NADH. Now, the acetyl-CoA or increased concentration of acetyl-CoA leads to the inhibition of pyruvate dehydrogenase complex. It inhibits pyruvate dehydrogenase, while on other hand, it activates the another enzyme, pyruvate carboxylase. What does it mean? The increased concentration of acetyl-CoA, if you look at over here, pyruvate, a 3-carbon structure is converted into acetyl-CoA, a 2-carbon structure, and CO2 and NADH are liberated. Now, what means or what is when the concentration of acetyl-CoA increases, it means that the cell has increased energy. Increased energy occurs when there are a lot of ATPs in the cell. This will lead to the inhibition of the downward processes of TCA cycle. So the concentration of acetyl-CoA will increase. This will lead to the inhibition of pyruvate dehydrogenase because the cell already has a lot of energy and there is no need to convert pyruvate into acetyl-CoA. And on the other hand, it leads to the activation of pyruvate carboxylase, which converts pyruvate into oxaloacetate, and this go on to develop into glucose or gluconeogenesis occur. If you look at the TCA glycolysis, a molecule of glucose is converted into pyruvate. During the conversion of glucose into pyruvate, there are 3 irreversible or regulatory steps. Number first is conversion of glucose into glucose 6-phosphate. The second is fructose 6-phosphate into fructose 1,6-bisphosphate, and number 3 is conversion of phosphoenol pyruvate into pyruvate. Now to go in reverse, or if we want to perform gluconeogenesis, we will need to convert pyruvate into glucose. But conversion of pyruvate into phosphoenol pyruvate cannot occur because the conversion of phosphoenol pyruvate to pyruvate is irreversible, and pyruvate cannot be converted into phosphoenol pyruvate directly. So for this, the pyruvate has to enter mitochondria, where it is converted into oxaloacetate by an enzyme pyruvate carboxylase using biotin as a cofactor, and CO2 is added into the pyruvate for conversion into oxaloacetate. A 4-carbon structure is formed. Then oxaloacetate can leave mitochondria and can be converted into phosphoenol pyruvate by an enzyme phosphoenol pyruvate carboxykinase. So this was all about conversion of pyruvate into acetyl-CoA and oxaloacetate. So if you look at number 2, pyruvate carboxylase, oxaloacetate can replenish TCA cycle or be used in gluconeogenesis. So pyruvate can be converted into oxaloacetate, which can either replenish the TCA cycle or can go on to develop into glucose via gluconeogenesis. So this was all about conversion of pyruvate into oxaloacetate and acetyl-CoA. Then we have another structure, lactate. One thing to remember is that conversion of pyruvate into acetyl-CoA requires two things. One thing is it requires mitochondria, and another thing it requires is oxygen supply, because once acetyl-CoA enters mitochondria, it enters the TCA cycle where NADH is generated, which enters the electron transport chain, which leads to the generation of ATP using oxygen. So for conversion of pyruvate into acetyl-CoA, we need two things, mitochondria and O2. So the conversion of pyruvate into lactate, it occurs through a specific cycle known as Cori cycle using an enzyme lactate dehydrogenase. As I have mentioned earlier, during glycolysis, glucose is converted into pyruvate. By doing so, we have increased concentration of NADH, which leads to the depletion of NAD+, which is required for the normal continuity of glycolysis. So what does Cori cycle do? It regenerates, or the NADH is converted into NAD+, which is regenerated to again participate in glycolysis. So lactic acid dehydrogenase, it is the end of the anaerobic glycolysis. So any condition or any state in which there is no mitochondria or O2 present, in these conditions the pyruvate chooses the Cori cycle, or the conversion into lactate for generation of NAD, which can be again used in glycolysis. The lactate dehydrogenase, it is the end of anaerobic glycolysis. It is major pathway in RBCs, WBC, kidney, medulla, lens, cornea, and Sertoli cells. So let's go through each of these structures. First of all, RBCs. RBCs lack mitochondria. So for normal glycolysis, RBC has to perform the conversion of pyruvate into lactate because they do not have mitochondria for conversion of pyruvate into acetyl-CoA. So thus RBCs convert pyruvate into lactate. Then we have WBCs. WBCs mostly perform, or their action is in inflamed tissue. In inflamed tissue we have low oxygen concentration. So there is low oxygen, so they have to convert pyruvate into lactate. Then we have kidney medulla. Again, kidney medulla have low concentration of O2, thus they have to convert pyruvate into lactate. Then we have lens and cornea. These are avascular structures. Thus, due to the avascular nature, they have low oxygen supply, and thus they also convert pyruvate into lactate. The unique among them are Sertoli cells, which generates, or in which performs spermatogenesis. And for normal growth of sperms, or the fuel of sperm is lactate. So Sertoli cells perform Cori cycle because sperms need lactate. So this was all about conversion of pyruvate into lactate. Now let's review how does Cori cycle occur. So Cori cycle can also occur in normal tissues as well. In normal tissues, when there is lack of oxygen or there is decreased O2 delivery, in these conditions, the cells, especially the muscle cells, they choose the Cori cycle. So what happens? The glucose stored within the muscle is converted into two molecules of pyruvate and ATPs are generated. The pyruvate is converted into lactate. Then the lactate is released into bloodstream. This lactate can go and enter the liver where it is converted back again into pyruvate, and from pyruvate it can go on to develop into glucose or perform gluconeogenesis using ATP, and thus this glucose can be again released into bloodstream to enter the muscle cells. So this was all about Cori cycle. So what is the effect of this, or what is the clinical implication of Cori cycle, or how does this occur? Suppose we have an athlete or a normal person. It goes to gym and perform different exercises like bench press and bicep curls, and suppose it is his first day. So after going to the gym, when he goes to the home or his body cools down, the patient feels soreness within his muscles. This soreness is due to the accumulation of lactate, because the muscles have performed the Cori cycle, or there is the accumulation of lactate. The lactate have decreased pH, and this decreased pH causes muscle cramps. And why does the muscles initially performs Cori cycle while after going to the gym for 2 or 3 months, 3 to 4 months, muscles doesn't feel sore? The muscle soreness is absent. Why does it happen? Now the reason is that after going to gym regularly, there are some changes within the muscle. We have increased number of mitochondria, as well as the sizes of mitochondria are increased. So after 3 or 4 months, when you go to gym, the increased NADH or the increased pyruvates are converted into Acetyl-CoA by entering into the mitochondria. Thus, there is no accumulation of lactate after going to the gym for more than 3 or 4 months. But initially, it is due to the decreased mitochondria, and the available mitochondria's TCA cycle and electron transport chain are saturated. Thus, pyruvates are converted into lactate. This was all about Cori cycle. Now, at last, we have conversion of pyruvate into alanine. And sorry, one thing, the LDH is an enzyme which performs the conversion of pyruvate into lactate, and it is elevated in LDH is elevated in different diseases, or clinical implication of LDH is it is increased in different tumors. It is also increased in MI, and it is also increased in hemolysis. These are this LDH which is elevated in these structures is actually this enzyme. Then we have conversion of pyruvate into alanine, and it occurs via Cahill cycle. And the enzyme which performs is the alanine aminotransferase. It requires vitamin B6 as a cofactor. The alanine carries amino groups to the liver from the muscle. Now, to understand it, Cahill cycle, let's first of all understand what are amino acids and what are keto acids. When amino acid loses amino group, when an amino acid loses amino group, it is converted into, or the structure left is the keto acid, the amino group. So the examples of amino acids and their, if I write over here the amino acids and their respective keto acids over here. So when an amino acid glutamate loses amino group, it is converted into alpha ketoglutarate. So alpha ketoglutarate is a keto acid of glutamate. Then we have aspartate. When it loses amino group, it is converted into oxaloacetate. Same goes to alanine. When alanine loses amino group, it is converted into pyruvate. Now let's understand Cahill cycle. This is muscle. So in muscle, when amino acid loses amino group, this is the amino group of amino acid. So when an amino acid loses amino group, it is converted into keto acid, and this amino group is when converted into given to alpha ketoglutarate, it is converted into glutamate. When alpha ketoglutarate accepts ammonia or amino group, it is converted into glutamate. This amino group is actually given to the alpha ketoglutarate from amino acid. Now this amino group is now then given to pyruvate, and thus pyruvate is converted into alanine. Alanine now carries the amino group of amino acid. Now this alanine can now enter blood and go to the liver. This is liver. In liver, alanine again loses the amino group, thus alanine is converted into pyruvate, and alpha ketoglutarate accepts the alpha ketoglutarate of liver accepts this amino group, and it is converted into glutamate, and this glutamate enters the amino group of this amino group enters the urea cycle of liver, and thus amino group or the nitrogens are excreted from our body, and this alanine is converted into pyruvate. This pyruvate again converted into glucose via gluconeogenesis, and glucose is released into bloodstream, and again glucose taken by muscles converted into pyruvate through glycolysis, and the cycle repeats. So it was all about Cahill cycle. Hope you understand it. Now let's go through the enzyme pyruvate dehydrogenase complex. It is not a single enzyme, but rather it contains 3 enzymes. That's why it is called complex. It is made up of 3 enzymes or 3 subunits, E1, E2 and E3. The E1 subunit is known as pyruvate dehydrogenase. The E2 subunit is known as dihydrolapyl transacetylase, while E3 subunit is dihydrolapyl dehydrogenase. So pyruvate dehydrogenase complex is the combination of 3 enzymes. It is mitochondrial enzyme complex linking glycolysis and TCA cycle. If we go over here, the pyruvate generated through glycolysis is present in cytoplasm. So once it enters the mitochondria, it comes, this is mitochondria. So it goes through the outer membrane through voltage gated channels, and through inner membrane through specific carriers. Within mitochondria, the pyruvate encounters 2 enzymes. There is one enzyme, pyruvate carboxylase and pyruvate dehydrogenase. What does pyruvate dehydrogenase does? It converts the enzyme of one molecule of pyruvate into TCA cycle. And what is the reaction? By conversion of pyruvate into Acetyl-CoA, it requires CoA, and CO2 is released, and NAD is converted into NADH in doing so. So we have NAD, we have CoA, and the products are acetyl-CoA, CO2 is released, and NADH. It is regulated or differentially regulated in fed state, in which it is active, and in fasting state, in which it is inactive. The pyruvate dehydrogenase complex require 5 cofactors. I have learnt it that first 4 vitamins all are the cofactors for pyruvate dehydrogenase complex. Thymine pyrophosphate, which is generated from vitamin B1. Then we have B2 FAD, riboflavin. Then we have B3 NAD or niacin, and B5 CoA or coenzyme A generated from pantothenic acid, and additional cofactor lipoic acid. The enzyme complex is similar to the alpha-ketoglutarate dehydrogenase complex. It is another enzyme which requires same cofactor, similar substrate and action, which converts alpha-ketoglutarate into succinyl-CoA of TCA cycle. We will be going through, this is alpha-ketoglutarate dehydrogenase, which converts alpha-ketoglutarate into succinyl-CoA. We will be going through this enzyme in TCA cycle. So this was all about pyruvate dehydrogenase, the lovely coenzymes for nuts. This is the mnemonic for it, but I have personally learned it that first 4 vitamins plus lipoic acid. These are the cofactors required for it. Now, there is a poison known as arsenic which inhibits lipoic acid or the cofactor for pyruvate dehydrogenase complex. So what will happen? It will halt the glycolysis, or it will prevent the pyruvate from entering into the TCA cycle as well as the electron transport chain. So the clinical findings of arsenic poisoning are: imagine a vampire. So imagine a vampire, the mnemonic for it is a vampire vomiting and having diarrhea, running away from a cutie with garlic bread. So a vampire for pigmentary skin changes and skin cancer caused by an arsenic. Then the patient also have GI symptoms. It has cardiovascular symptoms like QT prolongation, and GI symptoms also have a characteristic smell of garlic smell or garlic bread. It is due to the oxidation of arsenic to arsenic oxide. The treatment for it is dimercaprol and succimer for arsenic poisoning. Now, how the pyruvate dehydrogenase complex is regulated. So what are the activators? Number first, we have an increased NAD plus NADH ratio. Then we have increased NAD plus NADH ratio, increased ADP. Both of these mean that we have decreased energy state of the cell. When the cell has low energy, under these conditions, the cell will have increased NAD to NADH ratio as well as increased ADP. So under these conditions, the cell need to convert pyruvate into acetyl-CoA so it can enter the TCA cycle and perform or generate ATPs. It also activated by calcium. Calcium is released during muscle contraction, which is also a trigger for energy requirement or the conversion of pyruvate into acetyl-CoA. And now, what are the inhibitors of pyruvate dehydrogenase? The first is increase in acetyl-CoA, the product of pyruvate dehydrogenase. If you look at over here, if we have increased concentration of acetyl-CoA, it will lead to the inhibition of pyruvate dehydrogenase. Why? Because the subsequent steps in TCA cycle all are inhibited by ATPs. ATP reflects the cell has high energy, so there is no need to convert acid, or there is no need to of acetyl-CoA to enter TCA cycle. So the pyruvate dehydrogenase is inhibited. It is also inhibited by increased ATP, which means that the cell has a lot of energy. This was all about pyruvate dehydrogenase complex. Now let's review mitochondrial disorders. These are group of disorders, and pyruvate dehydrogenase complex deficiency is one disease of a group of disorders known as mitochondrial disorders. So what happens in mitochondrial disorders? In mitochondrial disorders, the pyruvate cannot enter the mitochondria or perform the function of different enzymes present within the mitochondria. So one enzyme present within the mitochondria is pyruvate dehydrogenase complex. So it is deficient. Another disease is pyruvate carboxylase, present over here. So pyruvate carboxylase deficiency. Then we have cytochrome oxidase deficiency. which is present in electron transport chain. So all of the diseases of mitochondria, so what will happen to pyruvate? So pyruvate cannot enter the mitochondria, so it will be diverted into lactate. Let me clear the. So if pyruvate cannot go over here as well as over here, so it will go in this direction and in this direction. So what we will have? We will have the pyruvate gets, sorry. So what we will have? The pyruvate gets shunted to lactate via lactic acid dehydrogenase and alanine via alanine transferase. All of the diseases are X-linked, and the symptoms will be the patient will have lactic acidosis due to the accumulation of lactate as well as increased serum alanine starting in infancy due to the conversion of pyruvate into alanine via alanine transaminase. The patient also will have neurologic deficits as well as poor feeding and poor feeding and growth failures. So what is the treatment of these patients? The treatment is increased intake of ketogenic nutrients as well as vitamin B1 and lipoic acid. The function of or the use of giving the patients B1 and lipoic acid is that to optimize the remaining functional pyruvate dehydrogenases. So if the patient has any remaining pyruvate dehydrogenases, by giving thymine and lipoic acid and all the cofactors like first four vitamins and lipoic acid, so the goal is to optimize the remaining pyruvate dehydrogenases. And what is the function of ketogenic diet? Since the patient cannot convert, before going into further details, what are ketogenic substances and what are glucogenic? Ketogenic are those substances which are converted into acetyl-CoA or ketones, while glucogenic are those substances which are converted into glucose or TCA cycle intermediates. So glucose is converted into pyruvate. So we have to avoid those things which can be converted into pyruvate, because cell cannot convert pyruvate into acetyl-CoA and it will go into conversion into lactic acid or lactate, which will worsen the condition of patient. So instead we have to give such a diet which can be converted or directly into acetyl-CoA. So by giving ketogenic diet like high fats, the fats are converted into acetyl-CoA via beta oxidation as well as ketogenic amino acid, some leucine and lysine. These are an amino acids which can be converted into acetyl-CoA, which can enter the TCA cycle without going through the pyruvate dehydrogenase or pyruvate carboxylase. So this was all about pyruvate dehydrogenase complex deficiency. Now at last we have TCA cycle. It is also called Krebs cycle or citric acid cycle. The mnemonic for the intermediates is citrate is Krebs starting substrate for making oxaloacetate. While I have learnt it by the following mnemonic, can I ask super, sad, father, mother. Okay, so the very first intermediate is citrate, then I, isocitrate, A, alpha ketoglutarate, S, succinyl CoA, SAD, succinate, F, fumarate, M for malate, and O into oxaloacetate. So first of all, TCA cycle. The pyruvate is converted into acetyl-CoA or two carbon structure by pyruvate dehydrogenase. If you look at the reactions of TCA cycle, there are three reactions which are irreversible or the regulatory reactions. The first regulated reaction is or irreversible reaction is conversion of oxaloacetate into citrate or the combination of acetyl-CoA with oxaloacetate via citrate synthase. Then conversion of citrate into isocitrate is irreversible. The isocitrate is the mutated form, or the conversion of citrate into isocitrate occurs via citrate mutase. Then after Then we have again two regulatory steps or regulated reactions, the conversion of isocitrate into alpha ketoglutarate and alpha ketoglutarate into succinyl-CoA. Both of these reactions are regulated. The first step is regulated by an enzyme isocitrate dehydrogenase, where isocitrate, a 6-carbon structure, is converted into 5 carbon and 1 molecule of CO2 is released. Same goes to alpha ketoglutarate. The alpha ketoglutarate is converted into succinyl-CoA. A 5-carbon structure is converted into 4-carbon structure and CO2 is released. So these all are about the regulatory steps. The regulation occurs, the general rule is that when the cell has high energy, there is no need for TCA cycle. So high energy inhibits TCA cycle, while low energy activates TCA cycle. All of the regulatory reactions of TCA cycle can be understood or remembered by the simple general formula. So conversion of oxaloacetate to citrate, the ATP, which reflects the high energy state, inhibit conversion of oxaloacetate into citrate. Thus we have increased acetyl-CoA, and which inhibits the pyruvate dehydrogenase. So you can see that high energy states reflected by increased ATP, acetyl-CoA or NAD inhibits PDH. Same goes to high ATP or NADH inhibits isocitrate dehydrogenase, as well as the high concentration of ATP, NADH and succinyl-CoA also inhibits the alpha ketoglutarate dehydrogenase. The alpha ketoglutarate dehydrogenase complex is same like pyruvate dehydrogenase, which requires the same cofactors as the pyruvate dehydrogenase complex. The first 4 vitamins B1, B2, B3, B5 and lipoic acid, and it is also made of the same 3 enzymes. If we look at the TCA cycle, from succinyl-CoA is converted into succinate, and CoA and GTP is released. Succinate is converted into fumarate and FADH is released. Fumarate can be converted into malate, and malate can be converted into oxaloacetate. These all are reversible reactions. And conversion of malate into oxaloacetate, we have conversion of NAD plus into NADH. And if we look at the activators, the low energy states activates it. The ADP, the low energy state, activates the isocitrate dehydrogenase. Same goes to the low energy states. ADP also activates it, and the calcium, increased calcium levels, also activates the TCA cycle, because the calcium signals or reflects that the muscles are contracted, so muscles will need energy, so this will all happen. So during Krebs cycle, pyruvate converted into acetyl-CoA produces 1 NADH and 1 CO2. So conversion of 3 carbon pyruvate into 2 carbon acetyl-CoA, we have 1 NADH as well as 1 CO2 being released. Then the TCA cycle produces 3 NADH, 1, 2 and 3. So 3 NADH are produced, 1 FADH, 1 FADH2 and 1 molecule of GTP and 2 molecules of CO2 per acetyl-CoA. This means that 10 ATP per acetyl-CoA to everything per blue glucose molecules are produced via TCA cycle. TCA cycle reactions occur in mitochondria. The TCA cycle is the reactions of mitochondria are the evolutionized reactions of the organism. So why they have advantage over the lactic acid cycle? During the Cori cycle or anaerobic glycolysis, we have only 2 ATPs generated, while during TCA cycle we have 3 NADH. 1 NADH is equal to 2.5 ATP. So we have 3 multiplied by 2.5. 3 multiplied by 2.5, we have 7.5. 1 FAD is equal to 1.5 ATP, so we have 1.5, and 1 GTP is equal to 1 ATP, so we have 1. So if we add all of these, it becomes 7.5 plus 1.59 plus 1, 10. So 1 acetyl-CoA can go on to develop 10 ATP, and there are 2 molecules of ATP acetyl-CoA as one glucose molecule go on to develop into two molecules of pyruvate, so it generates almost 20 ATP. So it is a superior reaction. Now there is one typical, or there is one concept which needs to be understood. As the acetyl-CoA enters the TCA cycle and the oxaloacetate is also the part of TCA cycle, and oxaloacetate can go on to develop into glucose. So why acetyl-CoA is known as ketogenic? Why ketogenic substances, or why acetyl-CoA cannot be converted into glucose while the intermediate of TCA cycle oxaloacetate can be converted into glucose? The reason is that there is no, when acetyl-CoA is added into TCA cycle, there is no net gain of carbon chain. The two carbons of acetyl-CoA are released during the TCA cycle, so there is no net increase in the carbon chain. So that, for instance, if oxaloacetate goes on to develop into TCA cycle, there is a net decrease in 4 carbon chain. But by adding acetyl-CoA, these are released during the regulatory steps and there is no net increase. So anything or any substance which increases the net carbon of TCA cycle will go on to develop into glucose. For instance, the succinyl-CoA, which can be generated by conversion of alpha-ketoglutarate into alpha-ketoglutarate is converted into succinyl-CoA by alpha-ketoglutarate dehydrogenase, and succinyl-CoA can be converted into succinate. This same succinyl-CoA can go on to perform heme synthesis. Another thing or interesting thing is that this is the same succinyl-CoA, the mnemonic vomit, the odd chain fatty acids, as well as the glucogenic amino acids like valine, methionine, isoleucine, and threonine. They all are converted into methylmalonyl CoA, which can be converted into succinyl-CoA. Thus, they can be converted into oxaloacetate, and oxaloacetate can be converted into glucose. So in this way, these substances, if we come to the first or the main reaction, these are the substances which can be converted into propionyl CoA and further into methylmalonyl CoA, which can be converted into succinyl-CoA. And this succinyl-CoA can be converted into oxaloacetate, and oxaloacetate can be converted into phosphoenol pyruvate, and the phosphoenol pyruvate can be converted into glucose. Now, one last concept to understand is NAD to NADH ratio. During conversion of glucose to pyruvate, we have increase in NADH. NAD plus is converted into NADH. And we have also studied that the pyruvate dehydrogenase complex is activated by increased NAD to NADH ratio. So this enzyme only works when there is increase in NAD plus. But conversion of glucose to pyruvate, we have increased in NADH. So no matter what the circumstances of cell are, we will always have increased NADH because cells perform glycolysis, and this reaction will be inhibited because there is increase. For the proper functioning or activation of PDH, we need increased NAD plus to NADH ratio. The reason is that there are two different pools of NAD and NADH, and they are differently regulated. We have cytosolic NAD and we have mitochondrial NADs. So pyruvate dehydrogenase is activated by the mitochondrial NADs, while the conversion of glucose to pyruvate generates cytosolic NADs. So it was all about pyruvate dehydrogenase and TCA cycle. Now let's review or go through some memory drills. Which cofactor is inhibited by arsenic poisoning? FAD, thymine pyrophosphate, lipoic acid, and coenzyme A. Pause the video and think about it, and the correct option is lipoic acid. Pyruvate carboxylase requires which vitamin? Biotin, riboflavin, niacin, or pyridoxine? And the correct option is biotin. Pyruvate dehydrogenase deficiency commonly leads to accumulation of citrate, lactate, succinate, or fumarate. It is lactate. In the pyruvate dehydrogenase deficiency, we have increased lactate as well as increased alanine. And at last, lactate dehydrogenase regenerates ATP, acetyl-CoA, NAD, or FAD. And the correct option is NAD. The LDH converts NADH into NAD+.